Bom dia a todos. Vamos iniciar a nossa segunda palestra do nosso ciclo de seminários, webinars Covid-19. Eu queria agradecer novamente ao professor Arben por ministrar a palestra para a gente. Lembrando aos nossos espectadores que a palestra vai ser em inglês, certo? As perguntas pelos chats podem ser feitas em português, e a hora que a gente for fazer as perguntas, a gente passa para o inglês, e o professor Arben vai responder em inglês também, certo? Hoje, quem vai apresentar o professor Arben é a professora Cecília. Cecília, fica à vontade, passo a palavra para você.
Muito obrigada, Maiara. Good morning, everybody. Today is a great pleasure to us have again the opportunity to listen to Professor Arben Merkoçi. Professor Arben Merkoçi, he is a career research professor and leader of the Nanobioelectronics and Biosensors group at the Catalan Institute of Nanoscience and Nanotechnology in Barcelona, Spain. Arben Merkoçi obtained his PhD at the University of Tirana in Albania, his hometown, in ion selective electrodes, and since 1992 he has carried out research as postdoctoral fellow and research professor at several universities and institutes around the world. I could cite here the University of Budapest, Universidade Politécnica de Catalunya, Universidade Autônoma de Barcelona, and New Mexico State University in USA, when he was working with Professor Joseph Wang. His research is focused on the integration of biological molecules and other species with micro nanostructures of the interesting design of novel biosensors. So Professor Merkoçi is co-editor-in-chief of Biosensors and Bioelectronics and the principal, and he also is a member of editorial board of several journals in the electroanalysis and in biosensors fields, such as Microchimica Acta and electroanalysis. Professor Arben has published more than 300 articles that got more than 20,000 citations and supervised more than 30 PhD theses. Besides that, Professor Merkoçi is co-founder of two spin-off companies based on biosensors, the Graphenia Lab, devoted to the graphene patterner sensors, and Paper Drop, that is devoted to the clinical diagnosis using paper-based device. So Professor Merkoçi is a distinguished research in the biosensors field, and for us it's so good and a great pleasure to have here today to talk to us about the detection of pathogens using these disposable and low-cost platforms as a paper-based device. Thank you so much, Arben. Now the audience is yours.
Thank you very much, Cecilia. Thank you very much, Professors Maiara, Daniel, for the invitation, and everybody who is here assisting this webinar. It's really a big pleasure. I'll try now to share my screen. So you can see my screen now?
Yes, it's perfect, Arben.
Okay, thank you very much. So I'm going to speak today about paper-based biosensors we are developing in my institute at Catalan Institute of Nanoscience and Nanotechnology in Barcelona. And as you can see here in this screen, I'm going to speak about very simple devices based on paper, plastic, that can be useful for different kind of application with interest for pathogens detection. So first of all, I would like to thank my previous collaborators from Brazil. So I am really very happy that I have been collaborating since many years with a lot of colleagues in Brazil, ranging from the current one, so Professor Cecilia de Carvalho, who is with us from Mackenzie, but also other colleagues that you can see here from Andrés Santiago, Luisa Mercante, Dr. Walter Cataneda, professor now, Everson Thiago, doctors, so recent PhD students, Jose Manuel Clausinho, Dr. Livia Florio, Dr. Umberta Queda, Dr. Fabio Caetano, Dr. Giselle Elias Nunez, Dr. Luis Humberto, in fact, was one of my first Brazilians who came in my lab, Dr. Charlene Regina Santos, so I wish I did not miss any other one, so really very happy to have these colleagues, now my friends, in our laboratory who did a really great job. And of course, I'd like to thank all my collaborators. So in my group, from different nationalities, you see almost 11 nationalities. Some of them are back in the group, some are in different places around the world, working as postdocs, professors. So I'd like to acknowledge their work. They have been doing all this work that I'm going to show. So their acknowledgement is for them. So just a little bit about my institute. Catalan Institute of Nanoscience and Nanotechnology is in fact a joint research center, and this is why you see here different logos. CERCA, Generalitat of Catalonia, CSIC, UAB. So we are belong to UAB also because we are situated in this campus, but also we belong to the National Research Centers Council, CSIC, but also to the regional one, CERCA, and of course to the government of Catalonia, who also has been financing our research. And since several years, we are part of Barcelona Institute of Science and Technology, where different research centers, excellent research centers, join together to increase the collaboration, to looking for synergies, and of course, increasing the visibility, which is really very good for the science and the collaboration we are doing. So this is where we are. So our institute is in this area. So this is Barcelona city, and this is where campus of Autonomous University of Barcelona, and our institute is there. So I put here also here different biomedical research centers, hospitals in Barcelona, because our center has a lot of collaboration with them for the biomedical research related collaborations. So being in a campus of international excellence, we have nice facilities. So in addition to the business area and technological park in the campus, near the campus is also the synchrotron, one of the fewest that are in Europe. So in the south of Europe, this is the only one that you have also in Brazil. Very nice one. I have been in Campinas. So, and we have nice facilities also in collaboration with microelectronic centers. So we have also this center next to us, and we also have some common facilities and also a nice micro and nanofabrication facilities with interest for our research. Our center is working in the nanoscience, nanotechnology, with a lot of interest in developing new science and new technology, of course, but also in technology transfer, where we are very much interested, public outreach, so to bring our science to the society, which is really very, very important. In our center, we have different research lines, so working in an area of nanoscience and nanotechnology. You can imagine this is very broad, and so we work from biosensors, for which biosensors and bioelectronics I am responsible. There is also another group of optical biosensors led by Professor Laura Lechuga. So we are two groups working in biosensors, and we have also a lot of activities and other colleagues groups working in nanostructures, synthesis, preparation, materials at nanoscales, microscopies, nanoelectronics, nanofabrication. So a lot of areas: nanomagnetism, photonics, quantum theory and simulation. There are also two groups: spectroscopy, spintronics, phase properties, and so on. So going a little bit in the topic of this talk, so we all know that there are several disruptive forces shaping the nowadays healthcare, from aging of population, chronic of disease, spending on health, pandemics, or the recent one we're living nowadays is requesting a lot of efforts from the research and technology to afford these pandemics. And in fact, conventional laboratories are working very well, and they can provide sensitive methods and techniques for analysis and information. They are very good also for basic research, but as you know, they have problems related to the cost, the time consuming. They are also, and of course, they request trained users and of course facilities for all these applications. And in fact, while these laboratories keep working and should work, there is a lot of need also for point of care diagnostics. And in fact, these are very, very important. And in this context, biosensors are very interesting devices that can be used as point of care for analysis, let's say, out of the laboratory. And in fact, this request for these devices, point of care, is increased a lot. And in fact, if we see to the market for point of care diagnostics, there are big numbers. So in this area, being blood glucose monitoring products, very, very important, but also other products like for hematology testing products, cardiometabolic pregnancy test is really very, very requested, in addition to infection disease and other applications. So looking to the biosensors we are working with, to be able to work as point of care, they should be assured. When we say assured, means affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, delivered. And this is really very, very important for these devices to be useful as point of care. And in fact, you know, the people working in biosensor, that this is a very multidisciplinary field, and there are a lot of biosensing technologies that can be offered depending on the kind of receptor or transducer these devices can use. And if we talk about receptors, one can consider from enzymes to DNA to antibodies, cells, and depending on the kind of transducer, then we can have electrochemical, optical, mass change, etc. So we can see this is really very, very broad, depending on the kind of receptor and transducer. So these devices can be of different technologies. And these are very, very interesting devices. I put here an example of very similar to glucose biosensor, but you can imagine that what we can do, how interesting should be if we can, instead of glucose, for example, detect cancer, detect other diseases, including COVID-related parameters, COVID-19 related biomarkers. So, and now, of course, with emerging of nanotechnology, nanomaterials, there are a lot of interesting achievements and developments, and these different nanomaterials, ranging from micelles, liposomes, metallic nanoparticles, quantum dots, to graphene, nanotubes, etc. Given their size and interesting properties, they can bring a lot of advantages and even new formats of biosensing, like wearable devices, smart packaging, implanted devices. So we are living in a period, the last probably 20 years, where there is an important change and improvements in the field of biosensors, with a lot of new technologies and new applications. And in fact, we can see biosensors nowadays everywhere, not only as medical devices, but also as wearable. In smartphones, there is a lot of interest to integrate biosensors, even smart TV, smart fridge, smart washing machine, smart houses, as food, plant control, for example, environmental monitoring, and of course also integration and use of sensors and biosensors in the future smart cities. So you can imagine how important is this field for a lot of areas, so everyday life. So we are going to have biosensors in the future everywhere. In fact, in our group, we have been working in this area since many years, and in fact, we are very much interested in development, for example, of new nanoparticles as materials building block for biosensors. We are very much interested in development and working with microfluidics, which is very important for pre-concentration, filtering, automation. But also, of course, we are very much interested in paper-based sensor, which is really very simple format. Simple is the best. We have been working and we have interest in nanomotors also to catch even single analyte and bring it to the sensing surface, and also a lot of interest in graphene. But I'm going to focus today mostly in paper-based sensors, so showing you some examples with interest for pathogens overall. So as I said, we are very much interested in use of nanomaterials, and in fact, we are very much interested in integration of nanomaterials into very simple devices with interest then for different application areas, being diagnostics one of them, but also we work a lot with environmental monitoring, food quality, safety and security, in addition to other industrial applications. So as I said, we've been working since many years in the use of nanomaterials, for example, reporting the use of quantum dots as new labels for immunoanalysis, DNA analysis, use of catalytic nanoparticles, for example, gold or core shell nanoparticles, use of plasmonic nanoparticles, nanopores for filtering and detection at the same time, carbon nanotubes, graphene, and then, of course, integration of these in microfluidics, use of nanomotors also. And what is very, very interesting, we have a lot of interest in combining these sensing and strategies, for example, in the case of diagnostic, combination of diagnostic with therapy in the so-called theranostics, or in the case of environment industry, combination of sensing of pollutants with removal of pollutants, so sensor removal technology, we call this. But let me show you about the topic today. So what we are doing about pathogens detection using paper-based sensors. First, I'm going to show you very shortly why paper-based nano-based sensor, what are the advantages, and how these operate, and how we can make these sensitive enough, giving some examples. And finally, I'm going to show you very briefly some works we're doing. So just ideas about SARS-CoV-2 detection with nanobiosensors, and finally some conclusions. So why paper-based nanobiosensors? So paper is formed by cellulose, is a low-cost material, easy to manufacture, and recyclable and biosustainable. And this is very important for its use, so having this material for our sensors. As paper has a porosity, so this porous matrix is very, very interesting because inside these pores we can do several reactions, but also the capillary forces create autonomous microfluidics. So we may have a running of liquids without energy, so this is a kind of zero energy device, which is really very important. In addition, paper is very simple to be tuned, so the architecture of paper can change very, very fast, just cutting it or doing some simple modifications, which are important, and this way you can change the microfluidics. Also in the paper, it is easy to integrate nanomaterials, so we can transform paper into plasmonic paper, but also fluorescent paper, putting there gold nanoparticles, quantum dots, and even other materials. In fact, working with paper is not new. Since decades, we have seen and we know people working in chemistry laboratory, for example, who are using pH paper. And the idea is, why not to use the same very simple format for other applications? So as simple as we do when we dip a piece of paper and look to the change of the color for the pH, why not to see other compounds, even proteins, bacteria, DNA, and this is what this field, so the paper-based sensor field, is trying to solve. So the use of these simple devices for the different other applications, And in fact, nowadays, these kind of paper-based sensors are well known as dipstick, LFAs, micropads, and in fact, the detection technologies are so broad, from optical to electrochemical, even chemiluminescence, MEMS connected to paper, and so on. So this is very, very interesting. And the most interesting is that now with the paper, with nanomaterials and nanotechnologies, thanks to the properties of nanomaterials, we can combine and have very nice platforms that can do very, very interesting detection, and then can be applied in different areas. Thanks to the use of nanomaterials with their large surface to volume ratio, their shapes, physicochemical properties, binding characteristics, that all combined in a piece of paper are really doing a very, very interesting job. In fact, it is called, just only for the gold nanoparticles applied in the paper, it's called like a golden area, where these different size, different shapes of gold nanoparticles bringing different plasmonics may be with interest for their use as labels in lateral flow analysis, which is something that we are also looking today, and it is reported in a lot of applications nowadays for COVID-19 biomarkers detection. In fact, working with paper-based sensors, lateral flow, is a very, very important hot topic in the research. And in fact, different kind of nanomaterials has been reported as plasmonic nanotags, for example, in paper-based analysis, where, for example, gold nanoparticles has been used as labels. We also have been working with these to have more sensitivity. Fluorescent nanotags, quantum dots, for example, also are used, and with these more sensitivity responses are obtained with interest for different applications. SARS, Raman-based detection methods using nanoparticles, and given their enhanced properties of SARS signal, has been also reported for in paper in lateral flow formats. We also reported the use of nanofibers, nanocellulose, as a matrix to increase the density of antibodies in a way that we can increase the density of antibodies, and this in turn can increase the sensitivity of the system. Paper-based sensors, lateral flow, for example, very easily can be connected to smartphone. This is also another advantage. So you can see here how the smartphone is connected and reading this lateral flow, or even using a Google Glass. So this is reported by Google, and this guy is observing here a lateral flow. So the images coming from the plasmonics of gold nanoparticles, he's reading with this Google Glass, and then the signal is treated and then transmitted to the office doctor or whatever. So you see how easy it is to connect these simple devices, even with smartphone, and make this very useful for real applications. When we work with these devices in general, so with all kinds of biosensors, it's very important to keep in mind the parameters that are key parameters, in fact, for their commercialization. So otherwise, your device, although very, very nice, cannot go to the market. And this is, of course, cost performance, so manufacturing capability and mass production capability. So these are well known for any kind of device, but we try to keep this in mind when we develop our paper-based sensors that may have integrated materials, integrated systems. So all these factors should be kept in mind to make possible these devices to go in the market. But how these devices can operate, and how we can make this device sensitive enough, so what strategies we can apply to make possible that a piece of paper can detect even proteins with interest for cancer or interest for different kinds of pathogens, where the detection levels, detection limits are very, very low, and the sensitivity should be very high. So starting from lateral flow, and I would like just to explain briefly for those who are not familiar with that, how these devices work. You can see here a format, a classical format of lateral flow. It is based on different kind of paper, different piece of paper with different porosity. the sample pad where the sample is introduced, the conjugation pad where the gold nanoparticles with antibodies are immobilized. Then we have this detection line with some antibodies that can detect the protein we want to detect, and then there's some secondary antibody that can detect with the primary antibody that can be used as a control line. So let me show you this video that explains very nicely what happened. So when the sample is introduced into this sample pad, the paper is serving as filter. So big particles like red blood cells, for example, white blood cells, they stay on top of this filter, these pores, and then the analyte is going around the platform. So by microfluidics, if we add also a buffer to help it, they run in automatic mode, so no need for energy. So the protein we want to detect, the analyte, is going to interact with antibodies that are on top of these gold nanoparticles and going through the fluid here, and they are going to interact with these antibodies here, so creating a sandwich like in a ELISA system. So thanks to that, the detection line is appearing, and then also these antibodies will interact always with the secondary antibodies here, bringing always gold nanoparticles as a control line. So afterwards, in a couple of minutes or maximum 10 minutes, the device will show very nicely what happened. So you can see here in these pictures different lateral flows that have a different control line. So you can see different red lines that correspond to the detection line with different intensity, and the same almost control line that shows that the system is working properly. So in this way, we correlate the change of the color with the change of the concentration. So depending, of course, on the format of the system. So there are different challenges in these devices, as I said before. So we want these devices to do sometimes more sensitive or sometimes multi-detection capability, doing these devices, of course, simple, low cost, and of course, connecting these also with Internet of Things through mobile phones and so on. So there are different challenges, and we're trying to address these in different ways. For example, to do these devices more sensitive, what we did is we connected the gold nanoparticles with some enzymes. In this case, we immobilized some HRP enzyme, and then instead of detecting the gold nanoparticles, we detected some byproducts of these enzymes using some substrates, for example, TMB, DAB, AEC, which are usually used in immunochemistry, so well known for some application in this area. So by using these modified gold nanoparticles, we are able to see much more sensitive signal. For example, you can see here from bare gold nanoparticles to the one modified with TMB, there is a clear change in the signal that you can see also in the change of the sensitivity here in this diagram. So in this way, we can use lateral flow with much more sensitive, even decreasing the detection limits. Instead of doing this, so using these gold nanoparticles modified with HRP enzyme, we also did some modification of the architecture. So as I explained before, simply by cutting the paper, we are able to change, for example, the sample pad or the conjugation pad. And you can imagine that when these changes happen in the geometry, the microfluidics is going to change. And in fact, we did some simple calculation. We modulated this, did some models of these microfluidics, and found out that how this was changing depending on the geometry. So in this way, we are able to tune the performance of the lateral flow based on the geometry of the system, which is in fact very, very simply done. So this is another way that we could, for example, extend or shorten the operation range, change, increase or decrease the sensitivity, detection limit according to the application. But instead of doing that, so changing the geometry, what we did is introducing some pillars, wax pillar onto the paper. So you can see here it's the same lateral flow, but now in the part of the paper, so in between the conjugation pad and the test and control line, we printed by wax printer some pillars, trying to find and optimize their distribution in a way that we can do the same as we did during the change of the geometry of the paper. So you can imagine that introducing these pillars into the paper, they are going to bother the microfluidics. So the microfluidics, in fact, is changing. So we are, again, trying to see the correlation between the number of the pillars, their distribution, upon the change of microfluidics, that is in turn changing the performance again. So this is also a very simple strategy that helps a lot in tuning the performance of the lateral flow according to the application. So changing the performance parameters, analytical performance parameters according to the application. And in fact, by doing this, we are able to detect, for example, even DNA. And in fact, we, in collaboration with a company here in Barcelona, we did detection of amplified Leishmania-related DNA using some amplification strategy, of course. You can see here what we do. In fact, we use this amplified DNA that was introduced into the paper, and by using some antibodies that were connected with peptides, we are able to detect directly this amplified DNA. And of course, by using some secondary antibodies modified with gold nanoparticles, we are able to amplify furthermore the sensitivity of the signal coming from these gold nanoparticles. You can see here, for example, what happened with the system without amplification. You can see just a few gold nanoparticles here in the detection line. But in the case of enhanced assay, where a secondary antibody with gold nanoparticles is used, a big accumulation of gold nanoparticles is appearing, as you see here from these images. And in fact, you can see clearly here the change of the geometry. In fact, we also use this here in addition to control system. We did also multi-detection with interest also for some control DNAs that are with interest for the specific diagnostics. In fact, this technology is in use also by our partners in Barcelona. So they are using in hospital San Juan de Déu, which is a clinical hospital with interested in the developments of some drugs for cancer, and we applied these during a European project to detect some different parathyroid hormone-related proteins as alternative to radioimmunoassay. So these simple devices that are very easy to be used has been applied also for this detection with interest for a certain type of cancer. So this lateral flow is quite versatile. So in addition to lateral flow, one can use also a vertical flow. And in fact, here we use this one. It was in fact by our Brazilian PhD student, Giselle. So she applied this with a shearing-like system where the paper with gold nanoparticles, as before, are now integrated in these two cartridges. One used as detection and the other one as a conjugation path. So the nice thing here is that the sample can be taken, for example, 5 milliliters of urine in the case of prostate cancer biomarker. And the nice thing is that the system here is used to pre-concentrate the biomarker. So at the same time, we do pre-concentration and we get the results immediately. You can see here the change of the color that corresponds to different concentration. So you can see the change, the increase of the color based on the increase of concentration of PSA. So this is really very interesting, and also can be used for preconcentration of, for example, virus in case of big volumes of samples. So this is a very nice application with interest for real application in different areas where you need to do a preconcentration of the sample. Another interesting material is paper, bacterial cellulose nanopaper. So I don't know if you have heard about this. This is very, very interesting. And this comes from a collaboration we had with Iran. So my collaborators, postdoctoral PhD students from Iran, brought this material to our laboratory. And this is very, very interesting. So multifunctional biomaterial with high hydrophilicity, high porosity, broad chemical modification capabilities, and high surface area. So what we did is we tried to load this material with different kind of nanoparticles, for example, with silver, gold, quantum nanoparticles, but also quantum dots, up-converting quantum dots that can be used for different kind of applications. In fact, we did different ways, either in situ production of these nanoparticles or ex situ. So produce these and then introduce onto the paper. So once we have this nanopaper modified with nanoparticles, you can see these are also transparent. These are nanopaper, bare nanopaper in front of the windows of our institute. So you can see it is almost transparent. This one is with silver and this one with gold nanoparticles. And you can see, of course, the gold nanoparticles, silver nanoparticles inside the network of this nanopaper. So to make some simple measurements, optical measurements, we need to set up some cuvettes, like two-dimensional cuvettes or micro-well plates, two-dimensional micro-well plates, in a way that we are able then to detect, like in a LISA, micro-wells, several paper and do some measurements. And in fact, we did some simple measurements with some compounds, like, for example, metimazole, iodide, cyanide, thiourea. Of course, we choose some compounds that interact with nanoparticles because they can probably, through formation of some complex, they are well known, they can corrode nanoparticles. And you can imagine that if you change the size of the particles due to the corrosion, the plasmonics of them will change. And you can see here clear changes of the color of the paper upon the change of the concentration. So we have a simple device that shows the concentration of these the different analytes. Of course, this is not selective because if we have these in presence of each other, they will interfere, of course, but we can do it selective. And in fact, I'll show you an example where we played with these and then using, in this case, in addition to quantum dots, we use graphene oxide as a quencher of the fluorescence. And we built a very simple system that is based on the quenching of the fluorescence by quantum dots when these are in contact with graphene oxide. For example, for the detection of bacteria, we use some antibodies against these bacteria. And you can imagine that in this case, where the bacteria is present there, the graphene oxide is interacting, of course, due to some electrostatic interactions, but the distance between the graphene oxide and quantum dot is large because we have like a few micrometer sized bacteria. So in the case of the sample that do not contain, that contains bacteria, the fluorescence is on. But in the case of the sample that does not contain bacteria, the graphene oxide almost approach the quantum dots, quenching the fluorescence. So we have a digital-like response in comparison to the analog-like response if you have a simple immunosystem with two antibodies. So this is quite sensitive and very, very interesting for safety security applications where you need to say just yes or no for a bacteria. And in fact, we put now everything into the paper. So all these components like quantum dots with antibodies and then run the sample and of course graphene oxide suspension. You can see here how it works, how simple it is building these devices. So we first prepared the paper-based sensor, set up in a paper of different porosity and building these different pads, of course. sample pads, conjugation pad. Afterwards, we introduce the quantum dots. So in this case, it is a cadmium selenide quantum dot introduced onto the paper. And after excitation, the photoluminescence is revealed. So these quantum dots show very nice photoluminescence that in turn then it is quenched when we introduce the graphene oxide suspension. You can see here how this photoluminescence is decreased upon interaction with graphene oxide suspension. And what we do is we control the change of the light and correlate this change of the light with the presence, absence of bacteria. So this is really very simple. Of course, after some optimization of all the components, we are able to quantify these bacteria, even in presence of other bacteria, in a very simple mode, as simple as we do, for example, with a pregnancy test. And in fact, now this paper-based sensor technology is in use by our spin-off company. So PaperDrop, who is trying to further develop and consolidate the technology in cooperation with other companies and research centers for different applications. So PaperDrop, in fact, is willing to apply and is applying this for different in vitro diagnostics applications, taking advantages of the technology, which is equipment-free, ultra-sensitive, low-cost, fast and easy. Of course, using an adaptive platform with a proprietary technology, so trying also to use some new kind of labels and also connecting these devices with the smartphone. So they're doing, the company is doing a lot of developments in cooperation with other fields. And finally, I'll just show you a few ideas about the work we are doing applying these technologies for SARS-CoV-2 detection using paper-based sensors, but also electrochemical biosensors. Very briefly, everybody nowadays knows what about the virus from analytical point of view for biosensing application. What is important for us is are two things. First of all, the genetic information, so the RNA it is inside the particle, but also the antigens, different antigens that are in the surface of this virus, like S protein, M protein in the membrane, in the envelope, the A protein, and the one that is in nucleocapside, so the N protein. So these different antigens in the surface of the virus have different properties, and in fact, the spike protein is the one that is very much reported and used so much for the detection of the virus. But also the N protein, which is nucleocapside protein called, is also very abundant, but it's important to open this membrane to take it. And in fact, this may be also another strategy to detect the virus. Of course, we should keep in mind the steric hindrance that this protein has if you want to detect this N protein, which is inside here. So, considering all this, there are different ways to detect the coronavirus. So either the direct detection of the virus particle using antibodies against these particles, but also indirect detection using through detection of human antibodies against this virus that, of course, are created once the virus enter in the body. So there are different strategies, and of course, all these clinical scenarios should be better should be understood very well. Otherwise, it should be difficult because in the body, once the person is infected, the levels of concentration and different kind of antibodies are generated. It is very important to understand what kind of antibodies you are going to detect, or if you are going to detect all these together. So in fact, there are different kits even offered using also cold nanoparticles. So it means that the development of nanotechnologies are already applied because all these, most of these lateral flows already contains gold nanoparticles, but also there are problems related to the sensitivity and also low or almost lacking of multi-detection capability. And this is what we are trying to address now, to develop devices that, based on the strategies I explained before, we can have these more sensitive and with multi-detection capability. So, and in fact, we are trying to build the devices that can have 2 detection lines in addition to control line. So we may be able to detect not only spike protein, but also an nucleoprotein that may be of interest to enhance the response if we are able to detect all this quantity of the antigen, which may in turn increase the sensitivity of the device and decrease the detection limit. In fact, as I said before, the detection of the antibodies generated once the person is infected are already reported. So recently there was a report on the multi-detection of IgM, IgG, so using for this SARS-CoV-2 infection. And in fact, these are already reported, but still more sensitivity is requested. And in fact, you can imagine that in paper, we can also integrate also detection of DNA, so using different layers of paper, for example, the layer with absorption path, of course, the one for extraction of RNA, in addition to amplification layer and so on. So possibilities exist to have more integration, which means, but of course, this will depend on the efforts, and we are willing also to address this depending on future projects. Another way we're trying to solve the problem is by using very high sensitive electrochemical detection. In this case, we're going to use impedance spectroscopy using silver nanoparticles based electrodes modified with some tile groups that can be, of course, connected with aptamers that may detect with very high sensitivity the whole virus, and then based on the change of the impedance and connected this with a smartphone, we can detect this as alternative to a PCR method that would request much more time. This is what we are doing. So as a conclusion, I'd like to summarize that paper-based biosensors are very, very interesting device. So in paper, we can integrate different kinds of nanomaterials and make these simple devices useful for different kinds of applications that can range from protein, bacteria, and even virus detection, with interest for different areas, for even, of course, for virus detection now or for future pandemics. So because you need to have very simple devices with cost efficiency, and of course devices like these that can be easily deployed in the places because there is a need for a lot of millions of devices that people any place in the world that can use these and without any restriction. So finally, I'd like to let you know that if you want to read more, you have written also very different reviews. So you can see here chemical society reviews on these topics, even for graphene, if you want to use these for different kinds of applications. And I'd like, of course, to thank all my colleagues. So we call this COVID-19 team, leading also by Professor Cecilia de Calvaño and Andrea, Claudio, Emily, Ruslan, and of course, Giulio, who are the senior researchers in the group, and other PhD students working very hard these days for COVID, in addition to other projects. And of course, my collaborators, our collaborators in Barcelona, in hospitals that will collaborate for real sample applications. Again, I'd like to thank all my groups and muito obrigado to all of you for following me. And thank you very much. I'll be very happy to give responses to any questions you may have. Thank you very much.
So thank you so much, Arben, for this great, amazing talk. It's very impressive to see the works that your group have been doing so far and how to use this very simple platform as a paper and transform this in very sensitivity biosensors. So it's really amazing. It's impressive. Thank you so much. So now you have several questions for you. You have so many people watching you from
I can
YouTube.
stop sharing.
Yes, you could stop sharing. Yes. You have more than 250 people watching you from the YouTube, and I will send some questions that you received from the YouTube chat. The first question is from Paulo Veiga Lima, the University of Ponta Grossa. He asked about these lateral flow devices, how these sensors will be accessible to everyone, or these sensors will be accessible only for a part of the population. In which country you think these sensors will be available more fast? And also if these sensors based on nanomaterials and also on paper device, you replace the conventional biosensors in the future.
Yeah, in fact, these devices, paper-based sensors, lateral flow, as I said, are really very, very cheap. But the objective we have in the laboratory is to build the technology first, to build proof of concept devices, and then these will depend on the technology transfer. So we'll need some companies to industrialize these. But I think that if these devices and also other research centers are building may work fine, that it shouldn't be a problem to bring these devices to any part of the world. And I think that should not be any difference between places, because we need to do diagnosis in the same quality in every part of the world. So, and then of course, sending to this part of the world cost-efficient devices, so devices that it should be easy for people to access and to use. I really believe that with some efforts, if all these technologies are going to work properly, then the pharmaceutical companies, through the help also of the governments and institutions, should be able to bring these devices to everyone, so without any difference, so everywhere, so no difference between countries, between people. So these are very cheap, and this is the right of everyone to be diagnosed in a very fast, efficient way. And the relation to the second question about these technologies, if they are going to substitute other biosensors, I think that all kinds of biosensors have the advantages and disadvantages, so it depends on the application. I believe that these are very simple devices for some application, but if you need for very, very high sensitivity, very low detection, there are also optical sensors. And in fact, in our center, we have a very good group, excellent group from Professor Laura Lechuga. She is building very high sensitive at the single level, probably analyte detections that may be useful for some other areas. So I mean, each kind of sensor should have its advantages. And I think there are application areas for any kind of biosensor. So the most important is to be robust, sensitive, of course, with the right performance according to application. I don't think that there is a universal biosensor for any kind of application. So this should be tuned according to the application.
Thank you so much, Arben. I will do one more question from the YouTube for you. So the question is from Cecilia Berin from the Federal University of Rio de Janeiro. Cecilia, she's asking if it's possible to integrate the Raman spectroscopy with these lateral flow these lateral flow devices. If this Raman spectroscopy could be a tool to do the read of the signal in this type of sensors.
Yeah, this is possible. In fact, there are some reports, people connecting simple Raman instruments that can detect even coupled with lateral flow. And in fact, we are not working with this, but people that are experts should take advantages if they are able even to build these simple readers, Raman, that can use, and they can take advantages also of nanomaterials that may have high Raman spectra, sensitive spectra, and this is really very, very interesting also.
Thank you, Arben. Now, we have some questions in the chat of the Google Meet. I don't know if Professor Tiago Paixão would like to do this question, open the microphone. You could be free to do this, or I can read your question, Tiago.
Thank you, Cecilia. Thank you, Abner, for the amazing presentation. And I would like to know a little bit more about the SARS-CoV and what are you thinking about? Is it interesting to combine, for example, electrochemical detection and colorimetric detection, like a multiplex method to detect the SARS-CoV? What are you thinking about this?
Yeah, there are possibilities, of course, to do combination, electrochemical, optical. Also, we are doing also and trying to do that. But I think it's very important to clarify and to be sure why we want to do that, because of course multi-detection is important, because in the case of this virus there are, as I said before, different scenarios. So we are very much interested, and there is need to detect the presence of the virus. But also there is need to detect the generated antibodies once the virus enter in the body, and sometimes it's important to detect different antibodies, because depending on the time of the infection, the body may have one antibody or other, or a mixture of them. So it means that you need to have, in the best of the cases, a system with multi-detection. But this multi-detection you can do even optical, just multi-detection optical, but also we can have this multi-detection combining optical and electrochemical. So this is why I'm saying it's important to see the advantages of using combination electrical optical, or probably you may do multi-detection with electrical or multi-detection with optical. So sometimes probably a simple eye-based detection, let's say optical detection, yes, maybe like a first screening to say that you have or not the infection, and then with electrochemistry you can have more sensitive detection to detect, for example, the level of immunization, or if you are interested to know how much virus load of virus you have, you go for, and you have this multi-detection system. So I mean, it depends on the scenario you want to apply this, but of course it's possible.
Thank you very much.
You are very welcome.
Thank you, Arben. You have now other questions from Professor Zeki now. Professor Zeki, would you like to do your question in person, or do you prefer I read your question?
Hi.
Hi, Zeki.
Hello, Zeki.
Hi. I wrote the question. First of all, congratulations for your presentation. It's very good, as always.
Thank you, Zaki.
And I was thinking about developing RNA aptamer-based biosensor to detect COVID-19. And I don't know, what do you think about the target? Which one would be best, spike protein, nucleocapsid protein, considering sensitivity and selectivity? Because I've seen some works, this property fight when you change the target.
Yeah, this is very important. As I tried to explain at the beginning, so if it's possible, one can detect RNA. In fact, what people are doing with PCR is trying to first, of course, extract RNA and transform RNA in DNA and then do the amplification. This is relatively long, but it is a standard protocol, and this is what is so far done. So people detect RNA through the detection of DNA, so transform the RNA to DNA using PCR, but this needs time, and sometimes normally it takes a couple of hours, and probably it may take 1 or 2 days taking the sample and so on. So this is long, but it is very, very sure and very stable as a result. But if we want to have something very fast, we should go for, as you said, probably if you are able to detect directly RNA, it should be fantastic. We are not working on that, but if you are able to do that, it may be very fast. But what most of the people and we are also working with is detection of the antigens. And in fact, spike is abundant from the one that are in the surface, and is the one that is mostly used because also its rate response is higher, and the generation of antibodies has been more successful. And most of the people are using spike protein, but the N protein, which is not on the surface but under, is more abundant. So, but if we are able to take this out, so opening this membrane and take this N protein, it should be much more sensitive. So, but you imagine, so depending on the possibility you have, you may play with one or the other. So we are trying, in fact, to see both. So probably we want to see at the same time S and N, because probably we may have more signal. So this is one thing we want to do. The other way is also to detect the whole virus through the detection of spike in electrochemical mode, using very sensitive device that I explained before, using impedance spectroscopy, and of course probably using graphene as a very nice surface that may have some advantages in terms of signal to noise ratio report. Yeah.
Okay, thank you very much.
You are very welcome, Zeki.
Thank you so much, Professor Zeki. Now you have a question from Daniel.
Hi, Arben. Thank you very much for this wonderful presentation. It was very nice. Regarding this SARS-CoV detection, we are seeing that we have a very big issue, that is the false negative response of some point of care methods. So how your sensor is performing about these issues about false negatives?
Yeah, this is a big problem in general in sensors, biosensors, and now, of course, in these COVID-related biomarkers, so false negative responses. So, of course, this may be different reasons. So, these, of course, first depend on the affinity, the quality of the antibodies, which is really very, very important, and other reasons. So, in fact, we are still not yet in the level of, we just started to work with the specific receptors, antibodies or aptamers, and of course we have been trying to screen first the different kind of antibodies that are in the market, and of course aptamers, and trying to find out which is more sensitive. So, and of course specific, selective. This is the first thing. As we are not experts in receptors, we just work with a commercially available one. But if I would suggest that for this work, if you have the possibility to work with experts that may prepare your own antibodies, your own receptors, or aptamers, for example, this is, I think, the best way to go faster, because you may be led by people that know what their products, the quality of their products is, how it is, and this is very, very important. Otherwise, we are going to lose time, as it happened also to us, checking different antibodies, and of course working with different blocking procedures, of course, of the surface, which is really very important, because this may lead to non-specific bindings or extra absorptions and so on, a lot of phenomena, very, very complex phenomena. We are still working on that, trying to solve the problems, because we still have issues, because as I said, we work with commercially available receptors, and we try to find the best ones that can have lower false negative responses.
Okay, thank you.
You're very welcome.
Thank you so much, Arben. We just have two questions for you from the YouTube. One is from Clara Marissa, from University of Rio de Janeiro, another from Marco Farias from University from Amazonas. Marco, he asking about how really work the colorimetric sensors. He has this question if it's possible to do the quantify analysis, or this is just like qualitative test. And the question from Clara is regarding about the errors or the standard deviation in this kind of sensors, in the lateral flow devices. how is it the standard deviation, the error between the analysis?
Okay, so in fact, with colorimetric sensors, for example, with these lateral flow devices, at the very beginning, decades where these sensors were in the market, they were only for qualitative at most or semi-quantitative measurements because the methods, it was only based on naked eye. But nowadays, when this can be connected also with smartphone, it's possible to have this totally quantitative, which is really very, very good. But of course, then it comes the other problem because depending on the quality of the technology fabrication, there are problems. So if you do not control well the fabrication of your sensor, of your platforms, of course, all the steps involved in the setting up of the sensors, ranging from nanoparticles, let's say gold nanoparticles, their modification with antibodies or preparation of all the conjugation step and all the detection and control lines. So there are a lot of steps that if are not controlled well, this may lead to errors, and of course standard deviation are high. And we also have had problems and try to solve these. But of course, at laboratory level, you can reach certain standard deviation that probably may go to 20, 30%, and sometimes still may be good. But if you want to do real application of this in the out of the laboratory, so a company should be involved, bringing much more reproducible fabrication techniques that will reduce all these errors, so as to have, as in any kind of device, much lower standard deviation. So it's a matter of course, controlling all the steps, and then, of course, the things are totally different from the lab to industrial fabrication, so which improve much more the standard deviation.
So, thank you so much, Arben. You are receiving so very nice feedbacks from the YouTube chat. People really loved your talk. So many good and nice messages. It's very, very nice. And also, I would like to open for Daniel or Maiara, if you would like to say something.
Yes, thank you. Thank you, Cecilia. Thank you, Professor Arben. It was an excellent lecture. Really, really enjoy it. And I would like to, I know Daniel have something to say. I think we can. Oh no, just I don't know. We have a little bit more time. Or we need to end. Yes.
Yeah, Arben, do you have time for one more question?
Of course, of course.
Okay. Yeah, Maiara, please go ahead.
I was thinking about when you saw the video of the lateral flow when you're cutting the paper. I was thinking how you could do it in a way that you can provide to the, at least not the whole world, but at least to Spain, all these types of sensors. How the process will, the fabrication process can become more. How can I say it? More
like a reproducible process?
Yeah. Of course, as I said, this was a typical example that you can use for probably in the laboratory, but if you want for industrial application, to go for other technologies, even for cutting, using even laser or some other techniques that can just like stamping, but they are cutting in a very, very fast mode. So there are different ways you can, of course, the industries may apply, but in a laboratory, you can use a very simple cutting. So just because you work with few sensors, you don't need thousands and thousands of sensors.
Yes.
So, of course, yeah. And we also are working also with laser for these.
Okay, so we can more portable.
Yeah. To do it more reproducible, of course. This is very, very important for any kind of device. it's sometimes the production way you have in the lab is totally different from the one that for industrial application is applied. So it means that you need something that ensure, as I said at the very beginning, some of the key parameters for industrialization is mass production capability, in addition to, of course, keeping the efficiency of the device and so on. So this is very important.
And your device, they have different layers of paper, right?
Yeah,
so how do you glue them? How do you connect the different?
Yeah, they come already with this glue, so they are already built in a way that you can connect this very nicely. So yeah. Yes.
Okay, so it's easy. Okay, Daniel.
Yeah, in principle, there are different possibilities to work with. Why not? So, and we are open to collaboration. So, with people that probably would like to collaborate with us, we are open to collaborate to using alternative material, always looking for advantages that probably one material may have in regard to the other. So, in fact, there are people working with different other materials. So, why not? Yes.
Good. I think that's it, right? I don't think we have.
Yes. We just like to say again, thank you so much, Arben. Your Professor Toro Murilo is sending regards to you, that your talk inspired a lot young researchers. That's true, it's a very inspired talk. And this is really good because you could see how we can use very simple materials that you can find these in our lab to produce very sensitive platforms for biosensing. Yeah?
Yes.
Yeah. Thank you very much. It was really a pleasure always. Of course, it was my first time to do this online seminar with Brazil, because I have been in Brazil several times, and each time I have been there, I have enjoyed the great country and great people. I will come again. So please feel free to write me, and you are really very welcome to come in Barcelona and to collaborate with us. As I said at the very beginning, the list of our Brazilian colleagues and friends is now long, and I wish it will keep maintaining because it's really a pleasure to have collaboration with Brazil. I'm really very excited always. Each new collaborator for Brazil is coming. Everyone has been great researchers, scientists, and great people. And I think that you will also enjoy Barcelona, Spain, a great country, great place to do research. So I wish you, I'll see you in Barcelona soon. And of course, let's hope that this pandemic is going, and we'll have soon come back to the normal life. But of course, paying attention to the distancing, social distancing, using masks because the virus is there, and we should pay attention to protect ourselves, to protect our people, our colleagues, our families, everyone. And I'm sure that we'll go and even stronger after this.
Okay.
Yeah.
Thank you so much, Arben.
Thank you very much.
Okay.
Have a nice day.
For you too.
Okay, bye-bye.
Bye-bye, guys. Thanks a lot for everybody.
Thank you.
Thank you.
It was really nice.
Obrigada a todos que assistiram.
Obrigado.
Gostaria de lembrar que na quinta temos uma palestra às 14h. E os links nós vamos enviar quarta-feira à noite. Obrigada a todos. E até quinta.
Tchau, tchau, pessoal. Muito obrigada.
Aqui ainda está passando. Ixi, parece que a última pergunta que você fez não passou lá no YouTube.